In this video, we will be reviewing the second part of Defense Against Disease for IB Biology. The topics covered include the role of T and B cells in immunity, HIV transmission and infection, antibiotics and antibiotic resistance, zoonotic pathogens, vaccination, and herd immunity. The activation of lymphocytes is a key component of the adaptive immune response known as clonal selection. Specifically, T lymphocytes recognize antigens presented by phagocytes. These T lymphocytes, or helper T cells, then activate B lymphocytes. Once activated by helper T cells, B lymphocytes proliferate through mitosis and differentiate into plasma cells. Plasma cells are antibody-secreting factories that produce large quantities of antibodies specific to the antigens of the invading pathogen. This clonal mechanism ensures a strong immune response, which will usually be able to eliminate the infection. Immunity describes the body's ability to resist infection from a pathogen. This can be achieved through the retention of memory cells, which are long-lived descendants of the activated B and T cells. Memory cells enable the immune system to quickly produce the required antibodies upon subsequent exposures to the same pathogen. Thus, immunity is the basis for long-term protection against diseases. Let's take a few minutes to discuss a specific example of an infectious agent, HIV. HIV, or human immunodeficiency virus, is a pathogen that causes AIDS. This virus is transmitted through bodily fluids, including blood, semen, vaginal fluids, and breast milk. Unprotected sex and sharing of needles increases the likelihood of transmission and are considered high-risk behaviors. For many patients, HIV often leads to the development of AIDS, the disease associated with this viral infection. Although there's currently no cure for AIDS, various treatments are available that can prevent or prolong its development in HIV-infected individuals. Without proper treatment, the HIV pathogen attacks and destroys T lymphocytes, more specifically, helper T cells. A reduction in helper T cells, or T lymphocytes, disrupts the cascade of immune cell interactions that ultimately leads to antibody production. Without enough helper T cells, B cells aren't effectively activated, so fewer plasma cells form and antibody production drops. This weakens the immune system over time, making the body vulnerable to other infections that would otherwise be relatively easy to eliminate. AIDS is therefore characterized by severe immunodeficiency. Antibiotics are chemicals that kill or weaken bacteria by targeting specific processes found in prokaryotes. These include preventing cell wall synthesis and inhibiting metabolic pathways. This specificity allows antibiotics to treat bacterial infections without harming human cells, which are eukaryotic. Unfortunately, antibiotics are ineffective against viruses because they are non-living and lack the metabolic pathways that these drugs target. Similarly, infections caused by eukaryotic organisms, such as fungi, are unaffected by antibiotics. The overuse and misuse of antibiotics have led to the evolution of antibiotic-resistant strains of pathogenic bacteria. In any population of bacteria, some individuals will be resistant to antibiotic drugs. When used improperly, these resistant bacteria survive and multiply, even at high concentrations of antibiotics, rendering standard treatments ineffective. Sometimes another antibiotic can be prescribed to combat resistant strains of bacteria, but the emergence of multi-resistant bacteria poses a significant public health challenge. To prevent these problems, it's important for doctors not to overprescribe antibiotics, and equally important for patients to complete their recommended courses of antibiotic drugs. Zoonotic diseases, by definition, originate in animals and then spread to humans. Zoonotic transmission, or spillover disease transmission, refers to the process by which a pathogen jumps from one species to another, particularly from animals to humans. This occurs when a pathogen, such as a virus, bacteria, or parasite, originally adapted to infect animals such as bats, pigs, rodents, cattle, and primates, encounters a new host species, humans, in which it can establish infection. Spillover is the process by which a pathogen jumps from animals to humans and can be explained in 3 steps. First, the pathogen, such as a virus or bacterium, primarily resides in an animal host, which acts as a reservoir. This animal species typically does not experience severe illness from the pathogen, allowing it to persist and potentially spread within its population. Then the pathogen is transmitted from animal host to humans. This can occur through direct contact with the animal, for example bites or scratches, through consumption of contaminated animal products, such as uncooked meat or unpasteurized milk, or via close contact with animals. Then, once the pathogen has infected a human, it may be capable of spreading from person to person. This secondary transmission can occur through various routes such as respiratory droplets, bodily fluids, or contaminated surfaces, depending on the pathogen, leading to outbreaks or epidemics among human populations. Spillover often happens in conditions where humans and animals come in close contact with each other, such as in wildlife markets, agricultural settings, or during activities like hunting or farming. Factors that facilitate spillover include changes in land use, increased human-animal interactions, environmental disruptions, and genetic mutations in the pathogen that enable it to infect a new host. Once the pathogen has successfully crossed the species barrier, it can spread among humans and potentially cause outbreaks or epidemics. Understanding spillover dynamics is crucial for predicting and preventing emerging infectious diseases. As mentioned, zoonoses are infectious diseases that can be transmitted from animals to humans. Examples include tuberculosis, rabies, and Japanese encephalitis. Zoonotic pathogens can spread to humans through vectors like mosquitoes or ticks, direct contact with infected animals, airborne or environmental exposure, or by eating contaminated animal products. Controlling the spread of zoonotic diseases requires a comprehensive strategy that takes human, animal, and environmental health into consideration. The recent COVID-19 pandemic is a prime example of a failure to prevent a zoonotic disease from spreading after being transferred to humans. One of the best weapons we have against pathogens, especially those of viral origin, is vaccination. Vaccines stimulate the immune system to develop immunity against specific pathogens without causing the disease themselves. They contain antigens derived from the pathogen or nucleic acids such as mRNA. Exposure to these antigens causes the adaptive immune system to respond as it would against any infection, producing memory cells that will be able to defend the body should it encounter the real pathogen later on. As you can see in the graph, when a person is newly exposed to a pathogen, the primary immune response is significant, but much lower than it is following a second or third exposure. This indicates that the immune system has retained memory cells from the first exposure, which results in long-term immunity. Global vaccination programs have been incredibly successful at controlling diseases like smallpox, polio, and measles. However, a lack of vaccination may lead to localized outbreaks of infectious diseases that are usually rare. Not everyone is capable of receiving vaccinations due to their age, health, or certain autoimmune conditions. For this reason, we can focus on the vaccination of a significant portion of the population in order to provide herd immunity to a given disease. The individuals with light blue have acquired immunity in the population. If the pathogen appears, the pathogen cannot pass freely among individuals and the chain of transmission is broken. The infected individuals have limited contact with susceptible people. So, if an individual is not immune and the population is, there is little chance that individual will become infected. Public health programs, education, and research are all needed to ensure vaccination rates remain high. It's important to understand that vaccine-related health research is ongoing and subject to change. The recent COVID-19 pandemic has underscored the importance of scientific literacy, effective communication, and evidence-based approaches to public health. Analyzing real-time data such as adherence of mask use and infection rates, like that shown here, can help to guide government responses and policy decisions worldwide. Thus, continuous evaluation of emerging data is crucial for managing ongoing and future public health threats. This video provides an overview of the key biological concepts related to infectious diseases and the immune system. By understanding these concepts, we can begin to appreciate the complexity of the body's defenses and the ongoing global challenges we face in managing the spread of diseases.